Thank you so much, Pinar. Really excited to be here with you today. We prepared two interesting talks. So the primer that I'll give here next will highlight two of our recent algorithms that we developed in the group, focusing on geometric deep learning and protein representation learning using protein interaction networks and then generative models for protein ligand design with a focus on improving protein target discovery. And then in the second part, in the seminar, together with Yepeng Huang and Owen Queen, two fantastic students in the group, will share some of the very recent work concerning multimodal protein language models. Okay, so throughout these two seminars, the primers will highlight various projects, code bases, data sets, various tutorials for those projects. Associated models are all linked from our lab website. So if you go to Zitnik Lab, HMS, Harvard, TDU, you will find many pointers to resources that will be highlighted throughout these talks. So with that, let's begin. We're all seeing how the scientific research is being upended and transformed now with the advanced use of AI models and recent development in large language models, generative AI, and other high-capacity deep learning models. And so recently we ran a poll with Nature asking 1,600 scientists around the world on their views on the rising use of AI and generative AI, particularly in scientific research. And of those that responded, 27% felt that AI tools would become essential to their field in the next decade. Another 47% felt that AI will be very useful for their field of research. And so what this shows us is that generative AI is already changing how we go about our everyday research activities by augmenting scientific search engines, by helping us draft or some documents or brainstorm research ideas. On the other end, there is another pillar of scientific research that is being enhanced and potentially empowered through AI. And this is now increasingly seen by AI being used to augment research, providing insights that might not have been possible using traditional methods just a few years ago. So in our group, we are specifically interested in laying foundations for AI that can help us advance the scientific understanding of medicine and therapeutic design. And the way we think of our projects is often that they map on this process of developing drugs and new potential therapeutic interventions that could help find cures or treatments for some of diseases that currently cannot be mitigated or treated well. So we generally start with some understanding of disease circuitry or disease mechanisms, either at the level of disease subtypes, or even individuals, and then are developing graph learning, multimodal, geometric deep learning models that are generating powerful representations of various elements of disease mechanisms in order to predict, to optimize, and identify potential candidate targets. And I will highlight in this talk one model of that type called Pinnacle that we recently developed. Once those protein targets that are identified, the big next question becomes, what are the leads or chemical compounds, small molecules, proteins, peptides, or other therapeutic modalities that could modulate the behavior of those targets by either binding to them, by indirectly influencing and exerting their therapeutic effects on those targets. And the way we're approaching that is by developing generative geometric deep learning models that operate at the level of atoms, residues, and interactions between those elements, and how those geometry is actually shaping in three-dimensional space. All that is done in the context of a specific relevant biology, often represented as relevant disease context, patient context, or cellular aspects. And finally, that leads to priority lists of candidate compounds, candidate protein targets that then feed into experimental screening, downstream experimental validation, and eventually matching of therapies to patient benefits. So we really go from molecular understanding of disease mechanisms and are developing models that can help us generate better scientific and medical hypotheses to make the drug discovery and development process more efficient. The purpose of this primer is to introduce you one model that is a graph learning model that can help with identifying greater protein targets. That's called Pinnacle. And then second, I will talk about generative models, PocketGen, that can then optimize the binding affinity between those targets and small molecule ligands. So let's start with the first part of the talk. And then the motivation here is kind of outside biology for a moment. So let's just look at the word apple. We all know that apple is a polysemic word, meaning that we can either grow an apple, or we can buy an apple. And the meaning of the word apple is resolved by sentence context. So we need to look into the surrounding words that, that the word apple occurs with or appears within a specific sentence in order to understand what is the meaning that was intended by the speaker or the writer when they use the word apple. Similarly, if we look at H2FX, H2FX is a gene that encodes for pleiotropic protein, meaning that its particular function is also resolved and can depend on biological context. Here, what we mean is the cellular context. H2FX and its product in an environment with olaparib would perform different function than the same gene product being included in the environment with a different chemical compound, meaning such as doxorubicin. So it becomes a question of how can we develop now AI models that would be able to dynamically adjust their outputs based on biological context in which they operate? We already know that that has been routinely done in natural language processing, in computer visions, in domain outside of biology. Wouldn't it be great that even in biology, we would have models that naturally take into consideration this biological environment or context within which we want the model to make prediction? And in doing so, the same model would be able to identify and detect that the role of H2FX and its products might depend based on the context within which we are studying H2FX and its products. So I will walk you through a model called Pinnacle, which is a contextual AI model that can support therapeutic research leveraging single-cell biology data sets to define biological context, and then learning powerful protein representations that are contextualized and adapted and dynamically adjusted to those contexts, which can then be powered or used for various downstream tasks but either directly using embeddings or fine-tuning them for a broad array of tasks that we will see in a few minutes, including using these contextualized protein representations to enhance 3D structural representations of proteins that are generated based on geometry of proteins rather than biological context. These contextualized representations can help us get more precise view into the effects the drugs might have across different contexts in different cell types and nominate targets in a cell type specific manner rather than in a cell type agnostic context-free manner. Okay, so how does Pinnacle work? At the very high level, the Pinnacle is a graph neural network algorithm that operates and is trained on graph structured data sets. So the data set that we'll be using here is a combined data set that consists of a single cell atlas, specifically tabula sapiens single cell atlas, together with a protein interaction network, and specifically the human reference interactome, where nodes indicate human protein coding genes and edges indicate interactions between the encoded proteins identified through proximity screens. So these two sources of data, the single cell multi-organ transcriptomic atlas and then the human reference interactome, are combined in order to build what we call cell type specific protein interaction networks. And I will explain in a minute how we build those. And once we have that integrated cell type specific network representation, we train and develop a new model that operates on this collection of cell type specific networks that are connected with each other based on cell and tissue hierarchies in order to learn cell type specific protein representations. So let's first talk about the data, and then I will talk about the model. And then after that, I'll discuss some of the results of Pinnacle. So the question becomes, how do we leverage single cell atlas and the human protein interaction network in order to build this multi-scale cell type specific data structure on which we can then train a graph neural network? So to be able to model proteins across cell type contexts, what we need cell type specific protein interaction networks. And so these are the networks that are shown here as these blobs or these small networks that you see on the periphery of this structure. And then what is the hierarchy or the connected structure that brings together these cell-type-specific networks is information about cell-to-cell interactions and known in hierarchical relationships about cell types being present in certain tissues and tissues relating to each other based on the tissue hierarchy. So the way we build these cell-type-specific networks that were shown on the periphery from the previous slide is that we start with a global reference protein interaction network. And then for each and every cell type from the single cell atlas, we extract a list of activated genes. And then we induce a connected subgraph of that interaction network based from the global interaction network. And that leads to a cell type specific gene network. Now we repeat this process for each and every cell type in the atlas. And this gives us a collection, a set of over 150 cell type specific networks, which are these networks on the periphery of the structure. The other aspect is this hierarchy, or this what we call a metagraph that connects cell type specific networks to each other. And to incorporate insights about cell type communication and tissue organization, we have built this metagraph that captures exactly these kinds of relationships. So the way metagraph is built is that we start with a set of cell type nodes. So for every context, which is a cell type context, we introduce its cell type virtual node. And then we also add nodes that represent higher levels of organization, which are broad cell types and then tissues. And then we add edges between cell types to tissues based on assignment of cell types and the tissues that those cell types were sampled from. Then we add information edges between cell type nodes based on cell-cell interactions, which we retrieve from cell phone DB database to capture the ligand receptor interactions between cells in the two corresponding cell types. And finally, we add parent-child relationships that relate tissue nodes to each other based on branded tissue ontology. And so what this process gives us, it gives us this multi-scale hierarchical cell type network structure that provides a unified data set on which a model can be trained. And this is now exciting because we have a consistent, unified, connected data representation, which can then directly be fed into Pinnacle model. So now that we have a better understanding of our data, let's talk about Pinnacle and how Pinnacle algorithm actually works. First, let me give you some intuition about the model. So our overall approach is that we have this set of cell type specific networks. Here specifically, we have 156 cell types contexts that span 62 tissues of varying hierarchical scales. And what we will do with Pinnacle is we will transform these networks into latent embedding space. And specifically what we want to build is we want to create a unified embedding space, so a single latent embedding space, where for each and every protein node from the input networks, we want to have a d-dimensional vector embedding in such a way that those vector embeddings of proteins capture the connectivity and topology of input graphs. What is important to note here is that the same protein might be activated in a number of cell types. So what we will get is not just one embedding per protein overall, but we will get a separate embedding for protein for each of the cell types that the protein is in. And the benefit of that is that we don't get a single integrated summary of a protein embedding that would entangle these various views, but we really had a more precise cell type specific representation of the protein. So effectively, that means that for each protein, we'll get a set of protein embeddings. And each of those embeddings in that set are specialized towards a specific cell type context. All those embeddings will be now in this unified embedding space. And the way this embedding space is being optimized, it's optimized through a self-supervised objective, specifically link prediction objective defined on the cell type specific networks in such a way that we say that for each and every cell type, say cell type C2, for proteins that appear as nodes in a cell type specific network C2, if those proteins are connected by an edge in that cell type, then we want their embeddings will be encouraged to be closer together in that region of the latent space that corresponds to C2 context. if the proteins in that C2 context are not connected by an edge in the input data, then their embeddings will not be encouraged to be close together in C2 region. And this is self-supervised learning, meaning that there is no explicit labels that are necessary around the functions or therapeutic targets when learning this latent space. Now, this is jointly optimized not only for C2, but also for C1, which might be a different cell type context, and actually for each and every of those 156 cell type contexts. We have this notion of repelling or attracting proteins in the latent space based on connectivity of input graphs. This process is repeated not only at the level of protein nodes in cell type networks, but also at the level of proteins to cell type interactions, and then cell type to tissue memberships, so that we get an organization of the latent space that reflects the hierarchy of a biological system, both at the level of each cell type, as well as relatively comparing cell types to each other and cell types to tissues. Okay, so very briefly, how do we generate contextualized representations mathematically? Since Pinnacle is a graph neural network, the way to train graph neural network is through a process known as neural message passing strategy. And the idea is to specify multiple layers of nonlinear transformations. Each layer is a form of graph convolution. And what happens at each layer is that the current set of embeddings, which are output embeddings from the previous layer, are being propagated or sent along edges of the input networks, and then non-linearly transformed to generate embeddings for the next layer. So now we have a slightly more complicated version of neural message passing strategy here. Why? Because we have not one network, but many networks, and there is this hierarchical structure to the way networks are built. Specifically, there are three key components to neural message passing in Pinnacle. The first component is that of using protein level attention within each cell type specific network, where effectively we're taking an embedding representation of a specific network, specific protein, say protein U, from one layer, and then look at the embeddings of its neighbors in that cell type specific network, and then based on embeddings of direct neighbors in the cell type specific network, then update the embedding of that protein. Now this gets repeated in every layer of neural network. The second important component is that of doing message passing not only within each cell type network, but also exchanging messages between different cell types. And the intuition behind doing so is that we want to capture and transfer knowledge from one cell type to the other, and we want to do that in a non-prescriptive manner, so in a learned data derived manner. And because of that, we have this additional attention mechanism that allows us to exchange neural messages between cell type to cell type edges and cell type to tissue edges. And finally, the third important component, which is here highlighted in blue, is concerned with exchanging neural messages between tissue nodes so that we can capture this higher level organization of the embedding space. Okay, so the overall, when this message passing process is complete and multiple layers of this message passing is done, that the final output of this passing procedure at the last layer are the embeddings, final embeddings of proteins. Those embeddings are fed into an objective function that is then being optimized through gradient descent, and this process is repeated many, many times. So what is the objective function that we use to optimize embeddings in Pinnacle? This objective function has three components. The first component is defined at the level of protein nodes. The second component is defined at the level of cell type nodes, and the third component is defined at the level of tissue nodes. So the level of protein nodes, the loss components has the loss function has two components. The first one is that of loss that encourages the model to learn embeddings that capture connectivity between protein nodes in the cell type specific network, and then also organize cell type specific regions in such a way that they are close to representation of the cell type itself. The second loss component is the one that captures relationships between cell type to cell type nodes and cell type to tissue nodes. And the third last component is defined as a link prediction between tissue nodes at this high level of structure. So finally, when we optimize, we use this objective function plus the neural message passing strategy and optimize Pinnacle model. The results of the trained model are this large number of protein embeddings. And for this specific data set, what we get around 500,000 latent representations of proteins. And altogether, those are representations that are contextualized to 156 cell types. What I'm showing here on the right part of the slide is just for visualization, UMAP visualization, purely for visualization purposes, how you can think about those embeddings. Where we see these 6 panels. For every panel, we highlight one specific region that corresponds of the latent space that corresponds to one specific cell type. So for example, in the top first row left, we see epithelial cell context. So every gray dot in this UMAP space is one protein. And then in this specific teal bluish green color, we show cell type region where proteins in epithelial cell context gets embedded. And then in the region, in the plot next to it, we show a specific different cell type context. And unlike various UMAP representations that you are seeing that generate embeddings of gene cell matrices, these are really embeddings of protein interaction networks across these cellular contexts. Okay, so why are we so excited about these latent embedding spaces? Not merely for the purpose of visualization, but we are primarily excited because these embedding models can now serve as some form of foundation or base model, where we can now take those protein embeddings across different cell types and either fine-tune them or directly query them or cluster them or do retrieval based on those embeddings and support a broad range of tasks. And so I will share with you a few examples of analysis and illustrate the power of Pinnacle. But broadly, unlike prior work, where there is quite a remarkable set of methods that are generating protein representations, the unique feature of Pinnacle is that it enables contextualized prediction across cell type contexts. And so what that practically means is that we can, with Pinnacle, support analyses that are concerned with multimodal deep learning, for example, where we expect certain cell types in biological contexts to be associated with ultimately disease or drug phenotypes. And we have ongoing projects in that area related to Alzheimer's and Parkinson's disease. It can support what we call context-specific transfer learning, because we can transfer knowledge from one cell type to the other. And it can help study the effects of drugs in a cell type specific manner to contextualize prediction. So in the next few minutes, I will focus mostly on this third use case that is shown on the far right of this slide. Okay, so what is the setup for contextualized prediction? So the broad or high level setup is shown here on the right part of the slide. We already know that we have a number of cell type specific contexts, and we will pre-train Pinnacle on that integrated network representation to generate the cell type specific protein embeddings. We have them, that's great. What we will do for contextualized prediction, we take those embeddings and we fine-tune them through a very simple, by tuning simple prediction module, with the purpose of predicting for each and every protein in each and every cell type, whether that protein, that cell type, could serve as a potential therapeutic target for a therapeutic area of interest. So the ultimate output that we want to get from contextualized prediction is we want to fill in this matrix where we have in the rows, we have different proteins in rows. In columns, we have cell type context. And we want to fill in this matrix such that a value at a particular position would indicate the score or predicted probability of the model being a good target for a specific therapeutic area in that cell type context. So in order to be able to fine-tune Pinnacle and specifically train this predictor model, what we need is now a small amount of labeled data that we will use to fine-tune contextualized embeddings. So the way we approach that is for a therapeutic area of interest, and I will show examples of that in the next few slides. We start with all druggable proteins that are in protein-protein interaction network, which you can think of as being represented by this gray circle. We then intersect that gray circle with the set of proteins that have been studied as consideration of being potential candidate targets for a therapeutic area of interest. And there is some set overlap that we expect to exist between these two sets of proteins. Then we can apply more stringent filters when we can look at not only proteins that have been explored as candidate targets, but proteins that actually serve as targets for novel therapeutic candidates that are currently in a clinical trial. And we can look at whether those therapeutic candidates have reached a specific phase of clinical trials, such as, for example, phase 2. And so what we get when we intersect all of these sets in the intersection of protein targets that that are targeted by therapeutic candidates that reach phase 2 or more and are also druggable in the PPI network, those proteins will serve as a positive training examples for training this predictor module. And then druggable proteins that are druggable but are not studied in a given therapeutic area serve as negative training examples to fine-tune this predictor module. There's a question.
Yeah, could I ask about how you define what a druggable protein is and how you identify them?
Yes, so the druggable protein here, proteins are defined as proteins that are known targets of any drug that is currently approved by FDA. And the primary source of data for us to be defining that very precisely is open targets. We're also looking into drug bank, a few other resources, but it's open targets and drug bank mainly to make these decisions. So we repeat this process that I just outlined for two therapeutic areas. So the first one is rheumatoid arthritis, and the second one is IBD, inflammatory bowel disease. So for each of these two therapeutic areas, we look at a set of diseases that would naturally fit under that therapeutic area, and then curate a set of clinical trials for each of those diseases, look at the drugs that are being investigated, and their candidate mechanisms of actions. And then this gives us a set of positive and negative training examples that we use to fine-tune pre-trained Pinnacle embeddings. And we fine-tune those embeddings with the purpose of finding and being able to nominate and study then the effects of those targets across different cell types and potentially predict new targets in a cell type specific manner. So here are the some of the results of this task. We can see on the left two plots, where the top plot shows performance of Pinnacle and state-of-the-art existing models for RA, and the bottom plot shows the same model being applied to IBD. So what we can see here is the following. So first, you can see that Pinnacle does not have just a single point, but multiple points. Actually, close to 156 points, because every purple point is here a different cell type context, because we can predict nominate therapeutic targets in a cell type specific manner, rather than what is predominantly used in published literature using methods such as various forms of graph attention networks, GATs, or multimodal network integration, illustrated here by a recent model, Bionic, that provide a single integrated summary protein embedding and therefore cannot produce contextualized representation. The interesting part here to note is that what we can see is that there are many cell types that are more predictive of targets, and there are some cell types that are less predictive of targets than integrated summary, which is integrated summary embeddings, which is not unexpected. It's not surprising, given increasing evidence and knowledge of certain cell types playing a more substantial role in the disease progression and then also treatments for a variety of diseases. And so that just strengthens the evidence that having a more cell type specific prediction can help us to be much more precise and potentially more accurate than just taking a kitchen sink approach across all cell types. In another interesting analysis that I want to highlight, we wanted to understand whether these protein level embeddings that are generated by looking at the global interaction networks plus single cell data provide information that is complementary to what we can get by looking at the embeddings of proteins that are generated by 3D structural geometry of proteins. And so what we have done is we looked at a handful of important protein interactions in immuno-oncology, specifically protein interaction involving PD1, PDL1, and another interaction involving B7-1 CTLA4. And what we have done is the following. So for each of those proteins, we took their 3D structure embedding generated by a model called Massif that generates embedding of those proteins based on the atom level representation of each protein and its three-dimensional shape of the protein. Now we take that embedding and we combine embedding of that protein with embedding of the protein generated by Pinnacle. And we take that embedding of the protein generated by Pinnacle from a relevant cell type context. In this case, those were specific immune-related cell types. And what we want to understand is whether these combined contextualized embeddings of proteins are able to better identify binding partners versus models, embeddings that would be generated based on structure alone, where there would be no contextualization. And we indeed can see that for these protein interactions important in immuno-oncology, bringing contextual information from immune-related cell types can help increase the margin between binding protein versus non-binders. So very exciting evidence showing complementary information that Pinnacle embeddings provide relative to structure, 3D structure embeddings. There's a question there. Yes.
So maybe you're actually about to get to it on this slide, but I was wondering on the previous slide if the cell types that you identified as most informative for prediction for RA and IBD, like what are the cell types all the way on the right? Is that okay?
Yeah, so that's a great question. And that question relates to, well, what are most predictive or informative cell types for a specific task? And this question is one of the most exciting analysis that we're interested in exploring further, because the way we can represent the results that we have seen here on the left slide, we can represent these results in the form of these ranked lists and ranked plots of cell types, where on the x-axis we order cell type contexts by their predictive power for a specific downstream task, in this case RA. And on the y-axis we show predictive performance of that cell type context for RA. And then that allows us to start nominating cell type contexts that are most predictive of a specific candidate target for RA therapies. What we can see here is that cell types that are deemed most predictive by Pinnacle give performance that is much, much higher than performance that can be achieved by using context-free multimodal network integration in the form of state-of-the-art model Bionic, which is this horizontal dashed red line, or what can be achieved using the reference protein interaction alone that is not contextualized cell type context. Now we can do the same type of analysis for inflammatory bowel disease, which we have done, and then we can go even one step further where we can look at one specific cell type and then look at what are the specific genes that the model deems most informative when generating that cell type embedding region, or when looking at a specific target, in this case ITGA4, and produce a ranked list of that serves as a form of priority list of cell types that are most supportive or least supportive of that gene serving as therapeutic target. So overall, what these results show, and many other results that I was not able to cover in great detail here, is that providing contextualized information to biomedical machine learning models is broadly useful. It's something that I would encourage more and more researchers to think about carefully in the field of how to build contextualized models. Pinnacle is an example of contextualized models. We broadly believe that models that can provide outputs that can be tailored to biological context will be essential also for a broader use of recent developments in foundation models for biology and molecular biology in particular. And once we have this contextualized ability to make predictions, we can support a broad range of tasks. So that was our recent work. Where are we now? So we are really thinking, we're developing Pinnacle with an eye of advancing Pinnacle in this line of research to ultimately create better links between single cell biology and fundamental research data sets, and then translational questions that relate to therapeutic development, leveraging single cell data sets. And so we believe that with the help of contextualized models, where we can contextualize prediction to a specific cell type, or even more broadly, disease subtype, or even individual patient, we can make much more better and more precise predictions to support therapeutic research. So our current follow-ups are to take the Pinnacle model that we discussed over the last couple of minutes and then do three important things. The first one is that we're training a much larger version of Pinnacle model. Some of in the audience might have noticed that Pinnacle, as I introduced, was initially trained on Tabula sapiens, which is a remarkable single cell atlas, but relatively small when we look at that atlas in the context of what is recently became available through CellXGene resource by Chan Zuckerberg initiative. So in addition to building XL Pinnacle model, we are also building disease-specific Pinnacle models, specifically PD Pinnacle, which is a version of Pinnacle focusing on Parkinson's disease, where we trained Pinnacle model not on a healthy single cell atlas, but on a midbrain, on a cell atlas that has strong representation of neuronal cell types. And third, RA Pinnacle that's specifically focusing on rheumatoid arthritis that is trained on a synovium single cell atlas with samples collected from patients that have rheumatoid arthritis. So just as an illustration, what XL Pinnacle means, it means that we scale the model from one data set of Tabula sapiens that has on the order of 150 to 70 cell types, depending on how the data is pre-processed and filtered, to a version to a Pinnacle model that would be trained on close to 80 million cells now, but at least 70 million after the data is being processed. And for the RA Pinnacle model that allows us to perform contextualized predictions at the individual patient level in relevant disease context, the main focus is on training Pinnacle model on single cell atlas that is collected based on samples not from healthy individuals, but patients that have disease activity index at different levels and are put on different types of targeted therapy for RA. So broadly, we think of these developments as developments that allow us to create then a family of contextualized models for single cell biology that can support the various therapeutic research, with ultimately supporting the kind of use cases where we generally start with some global base context-free inputs, then endow those context-free inputs with some notion of contextualized information, which can be that of single cell type context as we have seen today. And then Pinnacle model being trained on that joint context-free plus contextualized input to build a contextualized model that can support now a broad range of tasks. In the last few minutes, I want to change the focus a bit and go from building large-scale system-wide graph neural network models on multi-omic single cell data to thinking about generative models that, unlike representation learning models, can produce as output new designs. And specifically, the kind of designs that we're interested in is then designs that can help us understand a very precise atom-level interactions between proteins that can be identified by Pinnacle or identified through other means, and then potential small molecule compounds. And so I will just highlight one of our recent work, specifically PocketGen, which is a generative model that considered both the sequence and structural information to co-generate protein-ligand interactions. So it's a mouthful, but for the next couple of minutes, I will unpack what this actually means. So very conceptually, what is shown here on the left is the actual output and the process of training the model. So PocketGen can optimize and design both the structure at the atom level of protein. Specifically, it performs conditional generation on the area of the protein that interacts with the small molecule ligand, which would call here protein pocket, and then optimizes the structure of the small molecule ligand in such a way that the specific user-defined criteria, such as binding affinity, between the protein and the ligand would be maximized, and the specificity would be maximized as well. Generating such high fidelity protein pockets is quite challenging. It's still a big challenge for generative deep learning models in the field. There are numerous reasons why this is challenging, because there are complex interactions between ligand molecules and proteins that we want those ligands to bind to. There is lots of flexibility between ligands and amino acid side chains. And there are also complex sequence structure dependencies when we go from linear chain of amino acids to actually see how those fold into residues and what are the interactions between atoms in three-dimensional space. And so PocketGen is addressing these broad challenges. The way it operates is in the following. So we start as input with a candidate ligand molecule and with a protein structure. And in that protein, there is an area highlighted that is an area that we would want to optimize while keeping the rest of the protein as fixed as possible. So that would likely result in the generated design that has a higher chance of being actually synthesized and designed in the lab. So given this pair as input, PocketGen has two algorithmic modules. The first module is what we call is bi-level graph transformer. The second module is a sequence refinement module. And the purpose of these two modules is to jointly optimize both linear sequence of amino acids in the sequence space of proteins, and also consider additional constraints that are imposed on optimization when we look at the three-dimensional atom level interactions. This is a form of conditional generation because we condition this generative process on the binding ligand molecule, and we condition also generation on the rest of the protein except the pocket region. And the output of PocketGen is that it generates full atom level representation of the pocket structure, meaning both backbone and sidechain atoms. It also generates the residue sequence through this process of iterative refinement, and the ligand structure can be adjusted or not based on depending on what our user specifications. Very briefly, what are these two modules that I have mentioned just now? So bi-level graph transformer is, as the name implies, a transformer, a network that is trained on graph structured data. What's the graph here? The graph here is a graph of the protein and the graph of the small molecule ligand. These two graphs are built in such a way that they are built at the atom level. So we have nodes that represent residues, we have nodes that represent atoms, and we have edges connecting those different types of nodes based on interactions between bonds, bond lengths, the hedral angles, and other important properties of three-dimensional geometry. What's important to mention here that unlike graph neural networks that we have seen in the first part of the talk, which was Pinnacle, the neural network that we need to have here is a covariant neural network. That's the one that will take into consideration the fact that these structures, while they're very small, they really exist in three-dimensional space. So there are certain chemistry constraints and constraints that are imposed on the model in terms of the length of bonds or the hedral angles or planes that are taken into consideration by these models. And specifically, this bi-level graph transformer is focusing on modeling the structures. And it does so by having a two-level attention mechanism where one level operates at the level of atoms and the second level operates at the level of residues. The second component in the second module in PocketGen is sequence refinement module. Sequence refinement modules operate on sequences only without consideration of structure. So the sequence refinement module is, in fact, a protein language model that we adjust through LoRa adaptation by adding additional adapter layers into a protein language models. And those adapter layers are being fine-tuned during the process of training. So these two modules together, the bi-level graph transformer and the sequence refinement, constitute then PocketGen model that is then run through a series of iterative refinements where each refinement is leads to an optimized structure and sequence representation with the overall goal to maximize the consistency between sequence and structure-based view. Let's quickly look at some of the results of PocketGen. So first, we extensively benchmarked PocketGen on established molecular benchmarks. Here, this includes cross-docked and binding mod data sets. And broadly, what we see is that PocketGen generates protein pockets with higher binding affinity and better structural validity than existing models. Let me unpack that a bit. So first, how do we measure performance of the model? We adopt and implement a set of comprehensive performance metrics that consider both the predicted affinity between the generated protein and the ligand small molecule using Wiener docking score and also success rate. Then we consider local pocket structure metrics, where we are specifically looking at self-consistency root mean square deviation between the generated protein pocket and the predicted structure, a backbone. And then also look at predicted local distance difference test that reflects the confidence in the structure that is generated through ESM4 model. In addition to the local structural metrics, we also look at global metrics where we look at just the likelihood of that generated protein as a whole, meaning still having the relevant biological and chemistry properties. And that's done through by calculating a metric called designability and also using the SCTM score, which is the self-consistency template modeling score. We compare PocketGen against a number of state-of-the-art deep learning models and also physics-based models. This includes physics-based models such as PocketOpt and DPECT, as well as recently developed denoising diffusion models, including RF diffusion and RF diffusion all-atom version, which was published and available just a month ago now. And so what we can see is that when we look at generated designs by PocketGen, and we see that the model performs favorably across these deep learning as well and physics-based models and across various measures of evaluation that consider both the quality of the local global structure, the likelihood of designability, and the predicted affinity score. Another element that for consideration when benchmarking and developing generative models is a question of how many outputs do we need to generate in order to find one good one. That's something that perhaps as casual readers, you might just gloss over when reading exciting papers about generative molecular designs. But when you want to practically use these models, what happens is that these models can generate thousands, tens of thousands of designs starting from a single protein and ligand pair. So which now of those thousands, hundreds or thousands, should we then pursue further in terms of actual lab experiments? And so what we have explored and investigated carefully is the question of how quickly these designs can be generated, and second, whether those generated designs whether the number of generated designs is such that we need to generate many of them in order to find one good one. And so what we can see first is that PocketGen is 10x faster than many of physics-based models. And when we looked at predicted generated pockets that are prioritized by PocketGen as top 1, top 3, or top 5 candidates, we see a strong enrichment of those generated designs relative to what other models would provide. A separate interesting analysis also relates to the protein language model that I briefly mentioned as being an important part of the sequence refinement module. So what we can see is in line with the expected behavior in foundation models, when we integrate larger versions of sequence-based protein language models into sequence refinement model, the overall performance of PocketGen improves. So we can see that by this plot on the bottom right part of the slide, where on the x-axis, we show the model size of the protein language model measured in millions of model parameters. And on the y-axis is amino acid recovery rate, where higher amino acid recovery rate would indicate the better quality design. And we can see that integrating PocketGen with ESM models ranging from models that span from 35 millions all the way to 50 billions. We can see that integrating larger models leads to improved performance of PocketGen overall, which is an important result because it shows the promise of the model to be scaled up as larger base models become available, in this case, PLMs for sequencing refinement. As a very final point, just a few examples of how we are now applying PocketGens and using it to help design pockets of antibodies, enzymes, and biosensors for fixed target ligand molecules. And after convincing ourselves and establishing through rigorous benchmarking and multiple dimensions of model evaluation, that PocketGen performs favorably over existing generative models. We are now looking into using PocketGen to design interactions between cortisol and specific protein targets. And then a recent anticoagulant that was approved by FDA for patients with atrial fibrillation, APX, and then also as a way to design new biosensors that could detect certain molecules such as fentanyl in collected biosamples. And those are three very different use cases that demonstrate how PocketGen can support generative modeling across areas that are relevant for therapeutic development and more broadly bioengineering. So to wrap up this primer here, what I showed you throughout this talk are two recently developed models, specifically a contextual AI model, Pinnacle, that can predict and optimize and protein target study effects of drugs in a cell type dependent manner. And we believe that certainly this paradigm of contextual learning is really important in therapeutic development for a broad range of diseases and therapeutic modalities. And second, once protein targets are identified and further optimized, becomes a question of studying those targets not in isolation, but looking at those targets in the context of different types of chemical compounds and therapeutic candidates. And that requires modeling not only systems level or omics data, but also requires looking into biochemical structural representations of biomolecules and chemical compounds. And that can be done through models such as PocketGen and a broad area of models that are now being developed in this emerging, exciting field. In the second part of today's presentation, the seminar, as Pinar mentioned, there will be 3 speakers. I will not take more than 10 minutes or so of this time because I already gave kind of a seminar now, but we'll have really exciting results that we would love to share with you on new multimodal protein language models that we've been developing to provide a more holistic, functionally insightful view into protein functions. And so with that, let's start. This is the slide that we have already seen in the primer today, which relates to this notion of developing AI models throughout the drug discovery and development life cycle and developing different kinds of models that can help us generate better scientific hypotheses that can then support the design of new drugs and translating those drugs into safer and effective therapeutics. And one of more fundamental question behind the many of the research challenges and projects in that pipeline of the previous slide is that of understanding in a more comprehensive manner the functions and the roles that various proteins play. So we all know that proteins are workhorse molecules of life, and broadly improving our understanding of the functions and roles that different proteins have in terms of the biological pathways, molecular functions that are implicated in, how they can be targeted or influenced by different external perturbagens or different types of chemicals, and how they are associated with different diseases is really a fundamental question that powers lots of drug discovery and development questions, including, and this is mainly because the effects of drugs are often directly linked to the roles of proteins that can be either targets or even therapeutic candidates. So our broad goal is to develop these more comprehensive, functionally useful, functionally insightful models of proteins. And it's a challenging problem that it's at the frontier of lots of recent machine learning developments in biology, where there is a broad range of different types of language models being developed under this underlying joint paradigm or principle that we now refer to as foundation models. And the big idea of foundation models is to build a massive, consistent, unified data set that can be easily tokenized, and then train a large machine learning model, often in self-supervised manner, on that data set. And that model can then be trained once, but used many, many times to parameter-efficient fine-tuning adaptation for various tasks. And we have seen many successes of foundation models over the last several years, 2 to 3 years, across modalities, including natural language processing, computer vision, video understanding, multimodal learning recently. And underlying these models is the notion of language modeling, or sometimes called next token prediction, or colloquially we could refer to it as auto-completion objective, where the idea is that we have large, massive data sets of sequences, sequences of words in generally, or perhaps sequences of pixels, or we might have sequences of amino acids, and then we will hide parts of those sequences and train the model in such a way that it would be accurate, that would accurately fill in the missing parts. And that this language modeling objective, while initially developed for processing natural language sequences, in the last few years has led to quite successful development of protein language models that apply this principle to biological sequences, mainly amino acid sequences. Recently also DNA and RNA sequences. There are, however, several knowledge gaps in protein language models as they exist today. Those knowledge gaps are due to specific requirements that are unique to biology in terms of desired capabilities from the models, as well as due to relatively naive, straightforward transfer or adaptation of objectives, algorithms that were initially designed for natural language, for computer vision, to biology. So in today's talk, we will discuss 3 key limitations of existing models or knowledge gaps that we are excited about advancing. The first one is that of multimodality. Many protein language models today, prevailing approach is to train these language models on sequence data only. This is fantastic, wonderful, and remarkable, and it captures well evolutionary information. And to some extent, also functional insights can be derived from those models based on the functional content that sequence information provides. But what is lacking is a more explicit connection of functions, and also the ability of the models to incorporate additional data modalities when available beyond sequences. The second big challenge is that of generalizability. There's still an open challenge how we can take a single model and generalize it effectively through a broad range of different types of functional, disease, and therapeutic tasks. And then flexibility to make this model slightly more accessible to a broad range of scientists with diverse backgrounds, diverse type of interesting use cases, so they would be able to directly interact or query these models without necessarily requiring them to have coding experience. So I'll very briefly start with a kind of one porous man approach, or some of our initial attempts in this area, and then Owen and Yepeng will talk more about this. So one of our initial thoughts in this area was to improve language models in these 3 directions by looking at both the massive pre-training data set of amino acid sequences, and then also looking at 3D-dimensional structure of those proteins and those protein interactions. And so the way we approach that is by developing then a protein language model that combines the language modeling objective with the objective that is the link prediction objective on 3D structures to provide a slightly more functionally insightful view of proteins. And so a recent model called SIPT, structure-inducing pre-training, is a model that augments the language modeling objective with an additional term, which we call structure-inducing objective. And so what that means is that our SIPT model is then simultaneously optimizing both the language modeling objective through masking, and then also is generating this underlying latent space of proteins in such a way that those latent space is informative and predictive of interactions considering 3D structure of proteins. And this can be done very flexibly by allowing users to specify any type of pre-training graphs And what we show there through extensive benchmarking on considering those augmented PLM for function prediction is that by considering both the pre-training amino acid sequence data set combined with the pre-training graph and this joint objective function, performance of the model in terms of ability to predict functions accurately goes up significantly relative to what we can achieve if we turn off the pre-training graph and turn off the structure-inducing objective, which leads to a drop in performance from what is shown as blue curve on the right part of the slide, all the way down to yellow or even red line. And so this type of research recently now then replicated across different domains where we can flexibly consider as pre-training graphs not only protein interaction networks in this example, but we also can build pre-training graph in the form of literature associations based on scientific literature and incorporate that information in the model. Or in a different setup, we looked at higher order molecular interactions and built a different pre-training graphs. Across these three different setups, we are seeing quite consistent improvement in performance or stability, robustness, and generalizability of these augmented multimodal approaches. And so that has recently motivated us to start investigating this question much more thoughtfully and carefully by developing a novel multimodal language model that specifically focuses on addressing and filling in these three important knowledge gaps that I just mentioned. So I'll now invite Owen and Yepeng to join me here on the podium, and they will present this new model that they've been leading and developing over the last year or so.
Hi, everyone. I'm Owen, and I'm going to talk to you about one of our recent projects into trying to design a multimodal protein language model and how we evaluate that and how that's helping us build overall better models of proteins. Okay, so as Marinka very nicely sort of outlined, protein language models have given us quite a few advances here in the past few years, right? And so one of the big limitations with these protein language models is they're only trained on protein sequence information by definition, right? But one of the remarkable things is that by training on only sequence information, you actually see a lot of interesting sort of phenomena emerge by training with this very general sort of objective on these sequences. And so what the folks in the ESM paper sort of showed is that without any prior knowledge through just this mass language modeling objective on proteins, you actually get this sort of sense of function that emerges within these latent spaces of these proteins, right? And so much so that they're able to train this model called ESM fold that is built on the protein language model ESM and then can actually predict the structure of the protein from only the sequence, right? And this is remarkable because by training on libraries of millions of protein sequences, we get so much information that's encoded within these models that we can actually predict the structure, right? And so our question is, we want to think about phenotypic modeling beyond protein language models, right? There's so much information that's encoded within these models. However, us actually accessing the phenotypic information, we need some sort of modality to do that, right? And so our hypothesis is that we can use text to do that, natural language, right? And by using large language models, which capture phenotypic information that are encoded in biomedical text and databases, and sort of merging that in with protein language models, we can uncover so much more about proteins, but also facilitate really strong interaction between users and the protein language models that have been developed. And so this leads us to TXLM, which we're sort of calling this multi-scale universal protein annotation. So breaking down the sort of capabilities that Marinka outlined, so we have multimodality, flexibility, and generalizability. So we aim to sort of tackle all of these capabilities within this project, right? And so through multimodality, we are using a multimodal protein information. So we're using sequences, but also structure. So protein structure, using protein structural encoders. And then we're also using natural language models to be able to co-model text and proteins within our model. For flexibility, we want this model to be able to easily follow human instruction and interact with scientists from diverse backgrounds and levels of expertise with very little sort of coding experience at all. This is sort of the promise of the age of LLMs, is that we can use text and be able to interact with these models. And so we want to use that capability to facilitate interaction with the protein space rather than just the text space. And then also generalizability. So we want to be able to generalize to entirely new phenotypes in a zero-shot manner, and especially when protein annotations are scarce. And so we hypothesize that text sort of serves as a really rich modality for that, because you can encode any phenotype you think about in text, right? We write papers about phenotypes. We describe these things in written language. And so if we can encode that in our model, that gives us sort of endless levels of flexibility. And so how do we actually think about building the data sets that are going to train this model, right? And so I hinted at this fact that we want to be able to do multi-scale sort of universal protein annotation. So other folks in the past have tackled this problem of encoding proteins and text at the same time, right? However, a lot of these sort of approaches have only considered kind of molecular functions of proteins, right? So think like maybe uniprot, kind of like annotations that you can find online. And so what we want to do is we want to consider all the scales of lots of scales of biology at the same time. And so we want to consider not only protein and domain functions, so molecular functions. We also want to consider interactions between multiple different types of proteins. We want to consider the disease sort of implications of certain proteins and those relations, as well as the therapeutic use. So different drugs that interact with proteins, we want to be able to train our model to understand sort of all of these different scales. And so how we train our model is by using instruction tuning. I'll talk about what that is in a minute, but that gives us a unifying framework to be able to encode all these different types of relations under one sort of umbrella, and then we can feed it into our model as natural language. So some of the novel capabilities that sort of arise from this model that you don't get in traditional deep learning. So we have sort of free text description of phenotypes. So if you can describe it in natural language, it can be input to our model. We also, our coverage of phenotypes we claim is relatively universal because we're adopting that sort of representation from text. We also have explicit encoding of therapeutic modalities. And so you can explicitly sort of change the protein sequences that are input to the model. And then we have, we support things like compositional complex queries. And you can also depict context through both descriptions, so text, and also the proteins. And so just a bit of background, and then I'll explain exactly how this architecture looks, and then we'll dive into some results. So a background. So for folks that aren't familiar as much with the natural language space, there's this idea of what's called instruction tuning. And so instruction tuning, the goal here is we want to develop a foundation model that's easily accessible by practitioners. So how large language models are trained, you typically take, say, the entire internet, and then you're training next token prediction the whole time, right? And so your model's trained on just sort of trying to repeat text that's seen over and over, right? However, it turns out that the models, when you pull them, these base models are not actually great at following instructions. And so instruction tuning, the whole goal is you're actually giving the model an instruction that a human might give it, and then you're training it to sort of output something that follows that instruction. And so we see this as a really generalizable sort of framework, because what it allows you to do is it's an alternative to the pre-trained fine-tune regime that we've typically thought about within machine learning. And so in the past, you would have had, say, ESM. Some folks in here might have worked on these type of projects. We have ESM, we have some new distribution of proteins, we have some kind of thing we want to predict, right? We're going to tune the weights of ESM to predict the certain property of those proteins that I want, right? So that's a fantastic approach. The problem with that is that you would need to tune ESM on multiple different tasks to be able to perform each of those individual tasks. And so you can quickly see if you have limited data, right, that's going to be hard to do because you're not going to be able to tune those models quite as well. So the idea with instruction tuning is that you're going to train your model on multiple tasks at the same time, right? And then what you're doing is you're encoding your tasks as natural language, right? And so the idea is that your model can then perform zero-shot task generalization. So if you have an entirely new task, you can encode it in natural language. The model should be able to then generalize to that new task, right? And so this unified framework allows you to encode separate tasks together, right? And so you can encode, say, mechanism of action description and protein function prioritization as natural language, and it's under this unified framework. And so we use this sort of instruction tuning template that you see down here in the bottom right. And so we encode the task definition as, again, a sort of description of the task you're going to perform. You may give your model some in-context examples, such as this is some example of a task that you might perform. And then we give it the actual input that we want to query the model. So another bit of background is multimodal vision language models. And so VLMs have become very popular recently within the foundation model space. And the idea is that you want to interleave multimodal information into pre-trained generative language models, right? So you may have seen some of these models in recent conferences, such as Fromage, Lava, and more popular in industry. These VLMs are also becoming very popular in industry with models such as Gemini, right? Where they natively can handle sort of multimodal information. And so we derive a lot of our motivation for our architecture based off of this, because these models are able to sort of generate text, but they can also ingest images. So we think, well, instead of images, can we make that proteins, right? And then be able to have a model that has these wide capabilities, such as you see with a model like Gemini. Okay, so getting into the architecture of our model, right? So I'll follow sort of two lines here to show you two different types of capabilities that the model has. And then I'll describe sort of the broad sense of the capabilities that the model has from an input-output perspective. So one example of a query you might give the model is a protein retrieval query. So what you do is you bring maybe a custom library of proteins or amino acid sequences. You don't have to provide that. We can actually give you sort of, just a large chunk of uniprot and then retrieve from that. But that's optional. You can bring your own custom library of proteins, and then you can bring an input query. So you can say something like Alzheimer's disease is a neurodegenerative disease, ... right? I describe whatever sort of phenotype I want to query, and then you say identify a protein that's associated with this disease, right? So that's an example of a protein retrieval query. And then you have a phenotype generation query. And so what you do is you give the model a protein. So you give it a protein sequence, a protein structure. You say describe the disease associations of this protein, for instance, right? And then what we're going to do is we're going to give you text. And so let's follow each of these individual lines and see how our model is able to accomplish each of these. So first, for a protein retrieval query, right? So what happens is we take your custom library of proteins. We're going to pass it through a protein language model. We then have a projector that projects into a shared latent space that we encode in a vector database, right? And so in parallel, we're also taking your input query, we're tokenizing it, we're passing it to a large language model, and then we're getting embedding from the large language model that we're then querying into that vector database that you gave us of all those proteins, and then we're providing you a prioritized list of proteins that are related to your given query, right? And so that's sort of how the protein retrieval side works. So for the phenotype generation query, here what we do, so you can sort of input interleaved multimodal information, which is kind of in the weeds, but the idea is that you can insert as many proteins as you want into the input, and you can interleave them with natural language. And so that allows you to very naturally describe things such as protein-protein interactions. You say protein 1, you insert the information, and protein 2, you insert the information, for example. And so what we do is we take your sort of interleaved multimodal input, we decompose it into the textual parts and the multimodal parts. Multimodal parts get passed into separate encoders. And so for the sequences, we take that into a protein language model. For the structures, we take that into a structural encoder. We get embeddings for those, and then we perform this multimodal token composition, where we take the multimodal information, we insert it back into and interleaved with the text tokens, and then that all gets input to the large language model. Then we have the output of the large language model. The output is a distribution. And so what we do is we sample from that distribution to give you multiple phenotypic descriptions for your query, right? So this is something that isn't talked about as much if you don't work in the large language model space, but these models, you're sampling from them. And this is important for us because we all know that one certain protein might have multiple disease relations. And so by sampling, we can give you multiple different options for the output description or phenotype that you're interested in. And so this is sort of the unified umbrella, which we think about the TXLM architecture. So the important thing here is this is one model that can do all of this, right? So in the spirit of the foundation model sort of approach that Marinka mentioned, we have one model that is doing retrieval and also text generation, right? It's really a process of changing around how we process your inputs. We're still using the same underlying model weights. So we have one model that's trained to perform all of these tasks at once, right? And so TXLM supports this flexible task parameterization via textual task descriptions, right? And so some examples of some inputs we actually train our model on, right? And so the first is question answering or relation prediction. So the idea here is you give your model a protein sequence structure, and then you give it a sort of phenotype description. You say, are these related, yes or no? And then in natural language, we output yes or no, right? And we tell you whether these are related or not. So for example, you might say, here's a protein, here's a description of what obesity is this related to obesity, right? And then we say yes or no. The second is molecular retrieval and prioritization. So this is what we talked about, that retrieval sort of capability. So you can give your model some arbitrary textual input, and we can query proteins that are related to that. So for example, you can give it a composite query. You can say, here's a molecular function, here's a biological process, find me proteins that are related in both of these, right? And you can describe that in natural language, and we can give you an output list of proteins. The third is free response text generation. So you give the model a protein, and then you're getting text in the output, right? And so one example here that you can do is say mechanism of action description, right? So you can give your model a protein, you can give it a description of a drug, and you can say describe the mechanism of action of this drug on this protein, and then in the output, we give you a textual description of that mechanism of action, right? And so the important thing here, again, is this is one model that's doing all of this, right? And so by unifying everything under this sort of textual kind of domain, we can describe lots of tasks in a very flexible way. So how do we train this model? So I won't get too much into the weeds here, but we for these different capabilities, we have two main objectives. So we have language modeling, which is next token prediction, if you're familiar with how LLMs are trained. And then also we have, for the retrieval, we train that with contrastive learning or InfoNCE. The idea there is that in the shared latent space, you want proteins that are related to the query to be located closer to your output from your language model than proteins that are not related to the query. That's what helps us do this prioritization task with our vector databases. And I also want to mention that this is a very resource-intensive model. So this model is trained for about 1.5 weeks on 32 H100 GPUs. Our model size we're dealing with is about 10B, right? So 10 billion parameters. And we train this on the Kempner Institute GPU cluster. The Kempner Institute, if you're not familiar, is an institute over at Harvard. And so a big shout out to the engineers over there that have helped us set all of this up. So with that, I will hand it off to Yepeng.
Awesome, thank you, Owen. And now, before diving into our current results, let's briefly talk about our evaluation or benchmarking setup. So design the zero-shot setup to evaluate the model generalizability, where essentially we split the model. Can you see my mouse? Okay, great. And we essentially split, we split these annotations, which are protein phenotype pairs, by splitting the phenotypes. And these phenotypes are held out in certain thoughtful manner that we consider the structure within these databases. For example, in gene ontology, there's naturally this structure, this hierarchy between the gene ontology terms. And so we find out some subtrees held all of the gene ontology terms in the subtrees out. And in drug bank, there are these naturally ATC classifications of drugs. And so we also held out all drugs related to specific ATC classifications out. In this GenAd, we start with some initial seed held out diseases, and we held out all diseases that have certain significant overlapping protein associations with the diseases. And so these are essentially the hard setup. And even on these hard setups, our model, which is a blue bar across the, sorry, I keep clicking the slides for some reason. The blue bars, which is our model, consistently outperforms the other existing models, state-of-the-art models, such as biotranslator and ProTST, across different retrieval tasks in different knowledge domains. And here, we evaluate performance by comparing the concordance between the annotations prioritized by our model, given the phenotype, with the ground truth annotations. And we only show a rot here, but our model actually consistently outperformed the other models across metrics. So next, we further explore our model generalizability by comparing its performance across different splits. The normal split here essentially refers to a case where we simply split the annotations by its pairs without splitting the phenotypes. And the few-shot split is very similar to the setup we just described, zero-shot one. Despite that, you now put 2 annotations per phenotype to the training set. And so for the difficulty here, the normal split is the easiest one, and the zero-shot one is the hardest. And so as you can see here, the performance is stably strong for our model across these different splits, across difficulties, which demonstrate a strong generalizability of our model. In addition to retrieval, as Owen just outlined, our model is also capable of doing generation of text descriptions of proteins. Since the protein can be involved in various functions, drug mechanisms, pathways, and also diseases, you would want to condition this generation on certain knowledge domains that you want to focus on. And we can achieve this in our model via explicit instructions. Also, you want to sample the model multiple times, because each protein is also typically involved in multiple functions or diseases or drug mechanisms. And so in this first example, we see here is when we create the model, to generate disease-related descriptions for therapeutic. Well, in this example, for some outputs, the first two generative passages, I won't move my mouse again. The model is actually making predictions that reflect the evidence in the training data. And for the last two, the model is actually making novel predictions or generations, which some of them are validatable directly with direct evidence in literature, and the others are sort of having some indirect evidence support, but we need further validations for them. And in the second example, we can also attempt to generate function annotations for protein that are poorly characterized. Here we show this example of AKNAD1, which stands for AKNA domain containing 1. There's no function annotation for this protein across UniProt, Gene Ontology, or Reaptum as of yesterday when we made the slides. And so it's definitely not in our training data at all. And there is also no PDB structure, so we input a sequence and AlphaFold predict structure to the model. And the signal here we see in a generation of the model is actually quite remarkable, where when we condition the model to generate component-like, which stands for cellular localization, we see that the generation tests are largely reflecting microtubule-related, for example, cytoskeleton, and also spindles. And when we look at HPA, Human Protein Atlas, annotations of these proteins, which are not in our training data at all, we do find these two annotations of microtubules and also cytokinetic bridge for this protein. And when we condition the model to generate process-like descriptions, which largely is about the cellular process that this protein is involved in, it also generates some functions or processes related to microtubule-based movements, which is also something that the interacting proteins of this AKNAD1 protein is largely enriched for, where the interacting proteins are retrieved from strain. And lastly, we also validate that these generations are not just because of sequence similarity of the sequence with some other sequences, where the closest human Swiss Pro sequence similarity with this protein is actually about 30%. So this suggests that the model is actually performing some sort of reasoning under the hood, although we're still investigating these possibilities and also more capabilities of the model. Back to you, Marinka.
Okay, thank you, Owen and Yepeng. So that's our multimodal protein language model that we're currently actively developing. And as Owen and Yepeng mentioned, we have a version of the model trained and another one coming up very soon. And we're expecting to release this model fully open source and have a set of tutorials associated with it in terms of how to train these very large models in academic settings. There's lots of engineering behind the scene of how to train these models across many GPUs simultaneously. And these are really large models with billions of parameters that need to be loaded and tuned in interesting, novel ways, plus the new science that comes from providing functional disease and therapeutic annotations of the protein. So we're very happy to also engage with you, potentially collaborate or run some of your favorite proteins to our model. In the last couple of minutes, I would just like to spend a bit of time sharing with you some of the outlook and future directions for us beyond the protein language model and how we think that that type of models would fit into a broader range of data-driven models and that have been developed in biology over the last several decades. And so we were recently thinking critically about how the use of data-driven models have evolved over the last many years, starting from incredibly influential databases and search engines, going back to BLAST and other fundamental tools and resources that we all benefit from so much today, to then machine learning models that were generally trained in a specialized manner to perform well on a specific task and trained on a well-defined, narrow data set, to now, in the recent years, transitioning into more interactive foundation learning models that are not optimized to perform a narrow, a priori, user-specified task, but instead focus on a more broader range of tasks that those models can support. And so the question there is, okay, where is the future? Or what's next, or where are we now, and how we can think about building broader ecosystems of these type of foundation models that are now being developed for specific levels of biology. And we discussed today a lot proteins, but there are many other areas of biology that also are benefiting, and we're seeing lots of advancements in foundation model research in those areas. And so we are currently very excited about this new paradigm that is emerging on building AI agents as more versatile, flexible systems that combine different types of LLMs, that combine machine learning models, that combine standard databases and tools, including experimental platforms under a single unified framework. And so what that allows us is to think about start building companions or co-pilots that can support then everyday research that scientists do in the lab, at universities, and in other environments. So our outlook is really about kind of building this notion of AI scientists as multi-agent systems that can empower biological research with the ultimate goal of not only operationalizing a single data type, but instead we live within the broader ecosystem where human scientists can rely on different types of agents that can help them brainstorm research ideas, that can help manage and administer and process experimental platforms, and then also operationalize some of foundation models such as multimodal PLM that was discussed just now. And so this is all within this broader scope on the long-standing ambition for AI and biomedical AI in particular, in the development of AI systems that could make a major discovery on their own or relatively autonomously, in a way that that discovery would not be just a novel prediction, but it could really be a discovery that could potentially, eventually, at some point, be worthy of a Nobel Prize for filling the Nobel Turing Challenge. And this Nobel Turing Challenge, and the idea of having now models that can perform novel discoveries, is one of the grand challenges that has been also identified a few years ago. And we believe that the generative agents that can work and operate different types of models simultaneously can help support that type of initiative. What does that require? That requires not only building models, it also requires curating and building lots of data sets. As part of that, our group founded and currently actively develops therapeutic data commons that's available at tdcommons.ai. TD Commons serves as a meeting point between biomedical scientists and drug designers, drug developers who know what are the real bottlenecks in drug design and development. And on the other end, AI scientists, machine learning researchers who can design and develop powerful new algorithms once those bottlenecks are defined in a meaningful manner. And with the overall goal of supporting algorithmic innovation and advances in therapeutic science. And the commons come in the form of ready-to-use data sets, benchmarks, leaderboards, so we know what are the best in-class models for each of 20 plus therapeutic tasks, going from target discovery to predicting binding affinity, generating small molecules, generating large proteins, protein peptide binding affinity prediction, manufacturing, synthesizing, drug repurposing, toxicity, safety prediction, lots of tasks along the drug discovery and development pipeline and different therapeutic modalities, where for each of those tasks, we provide a mathematical, formal, rigorous definition of the task associated with data sets, leaderboards, and performance metrics to help us understand and benchmark the capabilities of now increasingly more powerful models, and then supporting a variety of different types of use cases that are both predictive in a sense, such as predicting biological targets, as we have seen in the first part of in the primer today, as well as generative outputs that either generate new phenotype descriptions or generate new molecular structures. And so with this type of ecosystems that consist of data and model hubs, then we are in the right position to start building generative AI agents and operationalize them in these multi-agent systems. And here are just 6 examples of the kind of collaboration schemes between agents that we are now developing that allow agents to interact with different machine learning models specialized for specific tasks, or foundation models that have very broad use, and then exchange their ideas, interact with each other, query each other through APIs, through mechanisms that allow them to learn how to use different types of tools and have a brainstorming session or have a roundtable discussion among 3 or 4 or 5 LLMs to solve a bigger complex question, decompose it into many meaningful tasks, and then build up the solution for those narrow tasks into a broader task. And so the future is really exciting, and I feel that this idea of building powerful foundation models that we can then operationalize and integrate within biomedical scientific workflows through generative agents can help develop AI-based scientists eventually capable of skeptical learning, skeptical reasoning that can empower medical research. So with that, we'll stop here and happy to take any questions. And as we have mentioned before, these AI models, datasets, papers are available at lab website, and therapeutic data commons is at DD Commons AI. And of course, many of these projects that we highlighted today would not be possible without support of funders, as well as a team of just fascinating, great, amazing students and fellows that work together towards addressing some of these challenges. So thank you for listening.